This video is going to be all about the prokaryotic cell wall. Now, we are focusing specifically on bacteria here. So we are looking at the bacterial cell wall in this video. So the cell wall provides structure and shape for the bacteria, and it does protect it from osmotic forces, and it also serves as an attachment site for flagella, which is going to be talked about in a future video, but it is one structure that helps bacteria move around in an environment. And the cell wall can also be considered a virulence factor. And a virulence factor is kind of like a weapon that bacteria or pathogens use in order to successfully infect a host. And we're going to talk all about that in Unit 3. But the reason the cell wall can act as a virulence factor is because some cell walls come equipped with an endotoxin, right? Toxins, not so good for us, but very advantageous for a bacteria. So more on all of these things later. When we're thinking about the bacterial cell wall, we can divide bacteria into two groups, gram-positive bacteria and gram-negative bacteria. And it is the structure of the cell wall that dictates which group a bacteria belongs to. Now, the key component of the bacterial cell wall is peptidoglycan, which is kind of a strange word, but if you look at the word, you'll notice that you can divide it into two parts. Peptido is like peptide, which is the name of the bonds that holds proteins together. And then glycan, whenever you see like glyco or glycan, think carbohydrate. So peptidoglycan is a protein carbohydrate structure. And so if you take a look at this picture down below, you'll see something called NAG, and it stands for N-acetylglucosamine, and you don't have to remember that, but that's going to be the blue circles, and the captions are covering up the name of the orange circles, but they're called NAM, and so we say that NAG and NAM are what build this peptidoglycan. Peptidoglycan is made up of NAG and NAM, repeating units, because if you look, it goes NAG, NAM, NAG, NAM, NAG, NAM, right, into multiple rows. But look at the green dots, okay? There are going to be some little amino acids in there, so that's going to be the protein component, okay? And so peptidoglycan has the carbohydrates and proteins in there together. But notice on the gram-positive picture on the left, you see these yellow circles, these pentapeptides that are holding the different columns of NAG and NAM together. And you'll notice that is not present in gram-negative, and that's going to be important when we go through the details of these cell walls on the next slide. Okay, so notice they both have NAG and NAM. They both have those little green circles, some of the amino acids, but what's different is the presence or absence of the pentapeptide. So let's go ahead and look at the gram-positive cell wall. So this picture shows you both, okay? So it's trying to compare them for you, but because there's a lot happening, let's first look at the left-hand picture, the gram-positive cell wall. So remember, all cells, no matter what, have the plasma membrane, and so that's this kind of middle region here. It's the phospholipid bilayer that you see. There's going to be a little bit of space in between the plasma membrane and the cell wall. That space is called the periplasmic space. You don't have to remember that term, but it's just letting you know there is a little bit of a gap between the border of the cell and the cell wall. What you're really looking at is what's above that periplasmic space. So you're going to notice that there are several layers of peptidoglycan. That's these blue kind of chains that you see here. So there's lots of peptidoglycan with the gram-positive cell wall, and that is good. Okay, peptidoglycan is a good thing. That's what's offering like that structural support and support from like osmotic pressure, things like that. So having all this peptidoglycan is awesome. Notice there's also something called teichoic acid that is holding all these layers together and kind of anchoring it to the plasma membrane, right? Because we want it to be firmly attached to the cell. So the two main features of the gram-positive cell wall that you want to remember is a thick layer of peptidoglycan and teichoic acid. Okay, so these are for gram-positives. Now let's look at gram-negatives. So again, just to kind of drill some of these words into your brain, notice, of course, because bacteria are cells, they have this plasma membrane, okay, that phospholipid bilayer. Again, there's going to be that little bit of a gap between the phospholipid bilayer and the cell wall. But now look at the details of this cell wall. Do you see how there's only one thin little layer of peptidoglycan? Okay, compare that to what you see with the gram-positive, right? There is significantly less peptidoglycan with gram-negatives, and that is bad, hence the negative. So it is bad or negative to have so little peptidoglycan. And so the gram-negative cells have to compensate for this. And in order to do that, they actually have a second phospholipid bilayer, which we call the outer membrane. Okay, so notice the gram-negative cell and the gram-positive cell are about the same size. Okay, but whereas the gram-positive has this really thick layer of peptidoglycan, instead, the gram-negative has a very thin layer of peptidoglycan with an outer membrane, which again is a second phospholipid bilayer. But there's a few little bonus features that come with this outer membrane, okay? And that is going to be lipopolysaccharide. Typically, I just call this LPS, lipopolysaccharide. And the reason LPS is a little bonus feature is because it is an endotoxin. Remember I mentioned that sometimes the cell wall is a virulence factor, a weapon the bacteria uses. Well, this toxin is that weapon, right? If we have a toxin going through our body, we're going to be weakened, okay? So LPS is associated with that outer membrane, but so are porins. Okay, so take a look where my mouse is. There's the porin, and you'll notice it's literally a pore, okay? So in order for something to access this gram-negative cell, it has to be able to go through one of these porins. Now, porins have a very specific shape. So what that means is only molecules that have a similar shape can get through. This is good for the bacteria and bad for us. And that's because if we want to try and kill a gram-negative bacteria, maybe we have an infection, okay, and we're infected with a gram-negative bacteria. We want to take antibiotics to get rid of it. But the antibiotics have to be able to go through these porins. So they have to have the correct shape. And that can be really tough, right? It's hard enough to find molecules that can kill a bacterial cell, but now we also have to make sure they have the correct shape so that they can even get into the cell, okay? So kind of a rule of thumb is gram-negatives are harder to get rid of than gram-positive bacteria. And that's because of the shape of the cell wall, okay? So to kind of recap here, the gram-negatives have a very thin layer of peptidoglycan. That's bad. So to compensate for that, they have this second outer membrane, the second phospholipid bilayer, which we call the outer membrane. And associated with that outer membrane is the endotoxin, known as LPS, lipopolysaccharide, and then these porins, which have a very specific shape and only allow very specific molecules to get in and out of the cell. So y'all, knowing the structure of a gram-positive versus gram-negative cell wall is really important. This is going to come into play later in the class when we talk about, antimicrobial drugs and other things, but also in lab when you're going to learn the gram stain and how we can visualize gram-positive versus gram-negative cells under a microscope. So please take some time to really be able to differentiate out gram-positive versus gram-negative cells. All right, y'all, that is it for this video. Let me know if you have any questions.